right, thanks. As an East Coaster, I refrain from thumbs up. But yeah, so this is a work that has been going back about 10 years or more with some collaborations, primarily led by Bernard Goffinet's lab at UConn. And so we're presenting a sort of benchmark milestone in this project of the systematics of the moss family Phaenariaceae. And so the full team, the lead author really on this work is Rafa Medina, who's at Augustana College in Illinois now. The funding came for Norm, John, and Bernard from the National Science Foundation. We also got some help from Yang Liu. And in general, the Moss Tree of Life team resolving where no one has resolved before, either at the very deep scale. We had a paper earlier this year on phylogeny of bryophytes. A couple of years ago, some transcriptomics, transcriptome sequencing in phylogenies, and then most recently, as of about an hour ago, a paper on Phaenariaceae phylogenetic systematics. Very good timing with this talk. And so, in general, these are talks that are united by a technique that is really exciting and really pushing the boundaries of phylogenetic systematics, and that is targeted sequence capture. And so our phylogenetic context, I don't have to convince the people in this room that mosses are cool, but kind of revising and revisiting some tricky old thoughts about how people think about mosses. So this is actually a quote from a book on mosses, and it says that mosses are true living fossils. And so that's just not true from a philosophical standpoint, and it's definitely not true when you consider all of the recent evidence that we have from a variety of different groups. We have evidence that the crown age of most moss genera is in line with most other plant groups, pretty young. Within that diversification, the spread of speciation can be quite rapid compared to the history of plants. This node is the ancestor of sphagnum, for example, at 14 million years ago. You have this bursts of diversification associated with a whole genome duplication events. So the genomes of these plants are changing really fast. So are the genes. We have different gene content that is present in a lot of our recently radiating moss groups. And another aspect is some original teaching when I was a student was that the molecular evolutionary rate in mosses was slow, and that is also not true. As we showed earlier this year, the rate of evolution in all three genomic compartments is equivalent or higher than the rate of evolution of similar genes in angiosperms. So all of that means that mosses are evolutionary dynamic, and they are definitely not primitive. Our extant mosses alive today are new. And along with that new molecular biology, there also needs to possibly be a reconsideration of how we attempt to put these morphological traits into context. And so we get rid of the primitive moss idea. We have these mosses, for example, that have embedded sporophytes. Traditionally, they've been all considered together in taxonomic groups. But there's also the possibility that these are all independent reductions of this trait. And so that's what this talk is going to be focusing on, whether this idea that we go from primitive to complex, or whether there is convergent evolution. And the focus group is the Phaenariaceae. So two classic mosses, Phaenaria hygrometrica with its tall, asymmetric, twisty capsule, or twisty seta with the capsule at the top and a nice peristome, shorter sporophytes of Piscometrium piriforme, and then a suite of species that have these embedded capsules. And a recent taxonomic revision tried to put Piscometrella patens, our model moss lab rat, into a genus that also included other species of plants with embedded sporophytes. So we have complexity, simplicity as the sort of taxonomic circumscription. So does that hold? And this matters from a taxonomic standpoint because of what I mentioned about convergent evolution. So if we imagine that there was a reduction of the sporophyte, so Funaria, Fiscometrium, Fiscometrella, and then a diversification of embedded sporophyte species, then we might be okay that Fiscometrella is a defined genus of diverse species. But if reduction occurred late in the development of Funariaceae, so that reduced sporophytes happen multiple places in the phylogeny, then Fiscometrella and the other embedded sporophyte taxa are polyphyletic, and that would indicate that the reduced sporophyte character is homoplagious, or a result of convergent evolution. Some early work on this was a few years ago in MPE using 10 Sanger sequencing loci, and it indicated that Fiscometrella, this is Fiscometrella reederi, Fiscometrella magdaleni, Fiscometrella patens, and Phaneregma are these examples of reduced sporophyte taxa, not monophyletic on the tree. But due to the poor resolution at these discrete loci, back then they refrained from doing anything taxonomically to Fiscometrella. And so we enter the age of targeted sequence capture, where we can take a genomic library where you just bust the genome into tiny pieces, that's like in black, and then we have probe sequences that we're able to hybridize with those fragments, and so we're able to pull out just the fragments that have the probes attached, and it would leave the other fragments behind. So it's a way to do high throughput sequencing to target a certain set of genes. In one example, we targeted all 120 organellar genes, and then we have another set of probes that targets 806 nuclear protein coding genes in Funariaceae. And so this presents a large amount of data, which is the focus of my research. And we put together the first paper by Rafa as the lead author on the organellar phylogeny. And this had some interesting patterns where, so it's all plastid and mitochondrial genes, 120 loci. And in general, we've got Fiscometrium in blue, Antostodon in red, in green, Funaria in red. And then these ones that are in black are reduced sporophyte species. And so they're scattered across the tree. Fiscometrella magdaleni, Fiscometrium reederi, Fiscometrium immersum, and patens, and Phaneregma are there. But one problem was resolution is still a little bit low, possibly as a result of this rapid radiation, all these branches suddenly appearing all at once in evolutionary time. And so we also have the potential hybrids detected where taxa are in the wrong place, in the wrong genus. Antostodon hungaricus is here, not with the other Antostodons. And then we have Fiscometrellopsis as well. So this possibly indicated there was hybridization going on in the Fiscometrium Antostodon group. So once again, failed to, or decided not to make any taxonomic changes until we had a nuclear phylogeny. And that's what we have generated in the paper that's out this morning. There's a lot of detail that I can go into, and I'm happy to talk about how targeted sequencing and phylogenomics works. Don't have the time right now. But for those who are interested, this was one really cool pattern that happened. What you're seeing, I don't have tips on the tree on purpose, but the pie charts indicate what proportion of the genes that we sequenced supported a particular arrangement of the species. And when we just use the protein coding sequences that we have okay support from individual genes. But by the nature of targeted sequencing, we end up capturing not just the protein coding part, but some of these regions that are next to it, usually introns. These are more highly variable and sometimes even longer. And so the proportion of blue, meaning supported in the pie charts, ends up going up on nearly every branch. So we're doubling the amount of data at the gene level, which really increases our resolution of this rapid radiation. And so another thing is that we are missing a number of species in our nuclear phylogeny due to allopolyploidy. These are taxa that are hybrids of two other Fiscomitrium or Antostodon species. And we identify these with our target capture data by aligning our sequence reads back to the sequences that we extract. So here are 3 Fiscomitrium samples that, so these colored bars are variation within Fiscomitrium. And so there's a lot of variation that's helpful for the phylogeny. But each colored bar only has one color. But this sample, most of the colored bars have 2 colors, which in the software is indicated as heterozygous. But we know that's bogus because these are gametophytes. And so the only other explanation is that they are allopolyploid, diploid gametophytes. And so this was true in this sample from Missouri, which actually seems to be related to the parent of Antostodon hungaricus as well. So things are complicated there. But we didn't see much evidence of Fiscomitrella having any of this hybridization, recent hybridization involved there. So that led to it being included in our large nuclear phylogeny. So we have, there's 2 main clades of Antostodon here. Fiscomitrellopsis africana is still down here and not a hybrid, and it has immersed capsules. Fiscomitrella magdaleni is up here within the core Fiscomitrium piriforme clade. Fiscomitrium reederi is sister to 2 clades of piriforme and the Asian species Japonicum. And then we have a Phaneregma and Fiscomitrella. Fiscomitrella patens does not have evidence of hybridization, but it does seem to not be related to any of the other species with immersed capsules. And so this means that the reduction in the CETA in the Phanereaceae is homoplagious, and these taxa that all have similar looking sporophytes do not form a natural evolutionary group. And so the article you can read today, it's actually part of a memorial issue that JSC is doing for Jochen Heinrich. And so from a taxonomic standpoint, Fiscomitrium patens existed well before Fiscomitrella patens. And so we are arguing for the abandonment of the genus Fiscomitrella, the reassignment of Fiscomitrium to Fiscomitrium patens from 1851, 2 new combinations of putting magdaleni and reederi, which were previously in Fiscomitrella, into Fiscomitrium. And so this creates a monophyletic Fiscomitrium, as well as abandoning a Phaneregma, which is a single species genus description. And so you may think that this is rather controversial, renaming a model species name. This happens all the time as we incorporate taxonomy and being more true to our understanding of evolution. We want to describe taxa on the basis of shared derived characters and not just hold on to names. The names mean something. And so this has happened in other taxa with some success. Some of our future work will be in full reorganization of the Phanereaceae, getting rid of a lot of other genera. We'd like to know, now that we know that it's convergent evolution, is there evidence that it's a separate mechanism to reduce the capsules in those different species? And since we have so much molecular evidence from the Fiscomitrium patens group, they will be able to help out with that. Identifying these parents of these hybrid taxa and studying the biogeography of polyploidy. Briefly on that, we have evidence that several species of Antostodon and Fiscomitrium are allopolyploid hybrids. And in the case of hungaricus, those are the 2 species, the 2 groups in red, and they are diverged, their parents diverged 25 million years ago. So it's a pretty extreme event. And our hunt for polyploidy in Fiscomitrium continues, and many of you in this room have contributed samples to us, and we're still accepting those samples from the US and Europe. And so you can check it out on iNaturalist, and we do in fact do stuff with your collections. This is a new postdoc on the project, Nikki Patel, who is processing them and sequencing DNA already. And so that's what we got. Thanks.